Caitlin Martin, September 6, 2022. This is my video for our lab on extraction of DNA for Genetics 1040. DNA is a molecule that all living organisms have in every cell of their body, apart from red blood cells. DNA contains instructions for the organism to develop, survive, and reproduce. The main role of DNA in the cell is for long-term storage of information. So I have already added my peas in with my salt water. I did soak my peas overnight in distilled water, and I'm going to go ahead and use my pestle to blend this up. I'm adding peas with the salt water to break the cells apart in the peas. So after grinding up my peas with my pestle, I put a cheesecloth over this cup and used it to strain out the solids from the liquids. I'm going to add liquid detergent to this, and the detergent has polar heads and non-polar tails, and these molecules will be able to pull apart the cell and the nuclear membrane, but leaving the DNA behind. So this is my soap and DNA solution. Now I'm going to add my meat tenderizer that contains enzymes that will cut the proteins that are covered and protected apart, but leave the DNA alone. So now that I've added my meat tenderizer and stirred it gently with my stirring rod, I can start to see the white DNA forming at the bottom. So now I'm going to add my cold 91% alcohol, and the alcohol is less dense than water, so it's going to float on top of the pea solution, and the lipids and proteins are going to prefer to be in the pea solution, while the DNA is going to rise and be in the alcohol layer. The cold alcohol causes a faster precipitation of the DNA, and it also helps in keeping the DNA intact and preventing denaturing. After adding my alcohol, this was my final DNA yield from this extraction of DNA lab, and I thought that it would yield more, and I thought that it would look more stringy. It is white, and when you look closely, you can see that it's actually stringing up from the pea solution. But when you kind of just look at it far away, it kind of just looks like a solid, and it has some bubbles at the top. But yeah, pretty cool experiment. I still see that maybe I have a little bit of DNA at the bottom that maybe hasn't risen to the top yet. So maybe if I let it sit a little bit longer, it would get bigger. But yeah, that concludes my extraction of this pea DNA.